Right. Good evening, everyone, and welcome to the week 5 live class session on the course on immunology. Okay. So, those who don't know me, I'm Srijan Sen from IIT Kharagpur, and today I will be taking the week 5 live class session. Right. So, I'm sharing my slides with you all. Yeah. So, I guess my screen is visible to you all. Great. So, fine. So, today's class is mostly on MHC, that is the major histocompatibility complex. Antigen presentation and antigen presenting cells. So previously we had talked about all of this. So introduced. So we have been well accustomed with these terms, right? Be it MHC or antigen presentation, APCs. So today we will go into a little bit of depth of what we had previously been introduced to. Okay. So, starting with the introduction and the immune surveillance. So, the core problem says, what does a T cell see inside a cell? So, T cells cannot recognize a free floating antigen. So, unlike antibodies, the T cell receptors only recognize short peptide fragments displayed on a cell surface scaffold. MHC molecules are that scaffold. So every nucleated cell continuously samples its own protein content and in specialized APC extracellular materials and displays fragments on the MHC. So there are two parallel surveillance systems. So the class 1 reports what a cell is making internally, and the class 2 reports what an APC has captured from outside. So this is how infected or the transformed cells gets flagged. So a virus infected or tumor cell that displays abnormal peptides on class 1 becomes a target of the CD8 plus T cell killing. So the entire adaptive cellular immune response depends on the display system. So what is being so the class 1 says, what am I making? That's the CD8 plus T cells, all the nucleated cells, and the class 2 cells, the class 2 reports like what did I capture? That is CD4 plus T cells, professional APCs. So what is MHC or HLA definition and scope? So MHC is the major histocompatibility complex. So it's a genetic region, the gene region controlling tissue histocompatibility discovered through transplant rejection studies. HLA is the human leukocyte antigen. So the human specific name for the MHC gene products, it was first detected as antigens on the leukocytes. Then there are three subregions by structure and function, the class 1, class 2, and class 3. So class 1 is the HLA A, B, C, that's the peptides to the CD8 plus T cells. The class 2 where you have the HLA DR, DQ, DP, these are the peptides to the CD4 plus T cells, and class 3 has no HLA genes, but it complements to C2, 3, 4, BF, TNF, the heat shock proteins that are present. So, a very brief history of the HLA discovery. So, in 1936, mouse transplant genetics, where Peter Gorer identified antigens that controls the graft recognition in mice. Then in 1954, Jean Dosset and Jean Van Root independently detected the antibodies against these leukocyte antigens in multiparous women and multiply transfused patients that led to the birth of serology. Then comes the serotyping era of 1960s to 1970s, where HLA becomes the central to organ and bone marrow transplant matching. And till today, it's a molecular genomic era. So crystal structures have been revealed. The peptide binding groove, the sequencing based typing reveals extreme polymorphism of how now 45,000 named alleles. So this is how the HLA discovery started, and where it is lying in today's generation. Okay. So the HLA locus lies on the chromosome 6, that is 6p21.3. So this is how it looks. So you have in the telomeric region you have the class 2, in the centromeric region you have the class 1, and it's subdivided into different categories. So class 1, it presents the endogenous peptides to the CD8 plus T cells, which are expressed on virtually all the nucleated cells. The class 2, which is towards the telomeric region, it presents the exogenous peptides to CD4 plus T cells, restricted to the professional antigen presenting cells. And class 3, where you do not have any HLA molecules, they complements the C2, C3, 4, and the heat shock proteins. Okay. So there is a exon intron organization. So the HLA class 1 heavy chain. So each structural functional domain of class 1 heavy chain is encoded by its own exon. So leader peptide, you have exon 1, alpha 1 and alpha 2 peptide binding domains, that is there in between exon 2 and 3. In the exon 2 and 3, then you have alpha 3 Ig like domains, exon 4. In exon 5, you have the transmembrane domain, and this way these things are well categorized. Okay. So this is the exon intron organization of class 2A, alpha, and the beta chains. So what you see that the alpha chain is divided into 4 exons, and the beta chain is then classified into another 6. Okay. So what do we need to know over here is that the alpha chain gene structure is basically composed of the distinct exons encoding the leader peptide, the extracellular domains, and the combined transmembrane cytoplasmic region. The beta chain, it features separate exons for the leader peptide, which is there in exon 1, then extracellular globular domains, the transmembrane domain in exon 4, and the cytoplasmic tail. So each exon block aligns directly with the functional segments of the mature heterodimer protein that is expressed in the cell surface. Okay. Now, HLA is the most polymorphic region in the human genome. So, from the bar graph, it illustrates the exponential growth in the number of officially named HLA alleles over the nearly 3 decades from 1998 to 2026. So, the exponential growth shows that advanced next generation sequencing discovers new genetic variants faster than the nomenclature committees that were officially name them. Okay, so the database statistics shows that as of early 2026, the IPD IMGT HLA database catalogs approximately around 45,000 alleles spanning over more than 45 genes. Then the HLA-B and HLA-DRB1 loci are the most diverse and are categorized as hyperpolymorphic. So the clinical challenges is that this extreme genetic diversity makes the donor recipient HLA matching for tissue or organ transplantation difficult. So there is rare or uncatalogued alleles further complicate accurate tissue typing. Now coming to the molecular structure of the HLA. So this is the architecture of the HLA class 1 molecule. So the key structural features is that it is a one membrane spanning heavy chain of 44 kilodaltons encoded entirely by the HLA-ABC genes located on chromosome 6. Consists of the three extracellular domains, a cytoplasmic segment and a transmembrane segment. Then they are non-covalently paired with B2 microglobulin. So it's a small non-polymorphic light chain encoded on chromosome 15. It is essential for the stable surface expression of the entire MHC class 1 complex. The alpha 1 plus alpha 2 domains form a closed ended groove, you can see from over there. So, this peptide binding cleft is flanked by two long alpha helices over a 8 stranded beta sheet floor, and it fixes and anchors the bound peptide at both the ends, where it accommodates short peptides of around 8 to 10 amino acids in length. So this thing is expressed in essentially all nucleated cells, where it serves as the universal self ID batch for the CD8 plus T cell surveillance, and it's notably absent from the mature red blood cells and largely absent from the central nervous system neurons. Okay, so coming to the architecture of class 2 molecule, HLA class 2. So it is also two membrane spanning chains. It has a heterodimer composed of an alpha chain and a beta chain. Both chains spans the cell membrane. So the domain organization, you have an alpha chain of alpha 1 and 2, beta chain of beta 1 and 2. The alpha and the beta are immunoglobulin-like domains located closer to the cell membrane, and alpha 1 and beta 1 are the outermost domains. You have the peptide binding groove, where it forms cooperatively by the alpha 1 and the beta 1 domains. Then because both the ends of the groove are open, so it can accommodate longer peptide fragments like 13 to 25 amino acids long, with ends protruding outwards. Now a little more about the closed and the open-ended groove and why the peptide lengths differ. So the class 1 groove is a closed ended, where it accommodates shorter peptides long anchored via the specific pockets. The class 2 groove, you can see it's open ended. So beyond the groove so it can extend beyond the groove at both the ends, allowing longer peptides, usually long. So it's because of whether it's a closed ended or an open ended system. So both the classes share a foundational fold of two long alpha helices, and atop of that an 8-stranded antiparallel beta sheet floor. This is a basic summary slide of about the classical genes that are there in class 1 and class 2, the composition of the chains that are mentioned over here, the peptide binding domains. Like for class 1, it's alpha 1 and alpha 2. For class 2, it's alpha 1 and beta 1. Then the groove geometry, which is closed at both ends for class 1, open for class 2. The peptide lengths, then the source of the peptides, which is endogenous or cytosolic for class 1. It can be exogenous and extracellular for class 2. The loading site is the endoplasmic reticulum and STAP dependent for class 1, and MIC endosomal compartmentalization for class 2. For tissue distribution, it's nearly for all the nucleated cells, that's class 1. And professional APCs, mainly the dendritic cells, macrophages, the B cells, okay, for class 2. Then the T cell partners, they differ, then the master transcriptional control. So it's a brief summary slide which you can look into. Now coming to a little bit more interesting things, which is the regulatory components, the elements in the HLA class 1 promoter site. So the core promoter elements, which is required for the basal level of transcription. So these elements are responsible for the baseline expression of the HLA class 1 molecules in almost all the nucleated cells, where you have the Tata box and the CCAAT box. So these are the standard basal promoter elements located near the start site. Then you have the site alpha enhancer B. So these sites bind constitutive transcription factors. So together with the Tata box and the CAAT boxes, they form the basal promoter shared with the most protein coding genes. And their main job is to position the RNA pol 2 at the transcription start site to begin baseline mRNA synthesis. Followed by you have some inducible regulatory elements. So while the baseline expression is steady, the HLA class 1 expression must increase drastically during an immune response to flag the infected or the abnormal cells. This is controlled by the upstream elements, the enhancer A. So this region is responsive to NF kappa beta, a major transcription factor during inflammation. We know that. The ISRE, that's the interferon stimulated response element. This sequence binds the transcription factors activated by the interferons. And so, in general, what you see is that the enhancer A and the interferon stimulated response element makes the HLA class 1 promoter strongly inducible by the inflammatory cytokines. And this serves as a key mechanism by which the infection or the tissue injury upregulates antigen presentation, alerting the killer T cells to the danger site. So for the so the regulatory elements and the class 2 enhancosome, so the SXY, or the, what do you call it, the ski module. So the enhancosome assembles on a highly conserved cis-acting regulatory region upstream of the MHC class 2 genes, known as the SXY module. It consists of 4 distinct sequence blocks. So the upstream most element of the promoter module, which is the WS box. You have the X1 box, which binds the RFX complex, the X2 box that binds the CREP protein, and the Y box that binds the NF-NAMA complex. So there are a specific set of transcription factors that binds cooperatively with the SXY module to form a stable nucleoprotein complex. So a heterotrimeric complex consisting of the RFX5, RFXAP, and the RFX A and K. So it specifically binds to the X1 box. The CREB binds to the adjacent X2 box, and then there is a trimeric complex that binds to the Y box. Then you have a master modulator, coactivator. So the role of the class 2 transactivator is the master regulator of the MHC class 2 expression. So the the CETA does not bind the DNA directly. Instead, it is recruited by the promoter through cooperative protein-protein interactions with the pre-assembled enhancosome. So once recruited, the CETA acts as the scaffold to bridge the enhancosome with the basal transcription machinery. It recruits key chromatin remodeling and transcription elongation, including histone acetyl transferases, chromatin remodeling complexes, general transcription factors, then the transcription elongation factors, and things like that. Okay, so okay, maybe we can go with a few questions, right? You can answer me in the chat box. So which chromosome carries the human HLA MHC gene complex? Yes, that is right. It is chromosome number 6. Chromosome 15 carries beta 2 microglobulin, the light class 1 light chain, which is not part of the HLA locus itself. B is correct. Which HLA class 1 component is enclosed outside the HLA locus on a separate chromosome? Very Very good. Yes. So beta 2 microglobulin is encoded on chromosome 15, which is yet it is required for stable class 1 heavy chain folding and the surface expression. Now, the peptide binding groove of an HLA class 1 molecule is formed by which two domains? Yes, the alpha 1 and the alpha 2. Hmm, right. Why does class 1 bind around 8 to 10 residue peptides while class 2 binds 13 to 25 residue peptides? Yes, because of the closed and open ends of those loops, of those grooves. So the class 1 groove is closed at both the ends, and the class 2 is open, so it can accommodate shorter or the longer peptides with their ends protruding for class 2. Right. Which factor is the master regulator that coordinates MHC class 2 transcription without binding DNA itself? Yes. The class 2 active transactivator, the CETA, which is recruited by this enhanceosome on the SXY module and coordinates the chromatin remodeling and the Pol2 machinery. So it does not bind the DNA directly. Yes. So is there someone trying to say something? I guess someone's microphone is switched on. That's fine. Let's go into the next part, which is the antigen presenting cells or the APCs. So, what makes a cell an antigen presenting cell? So, since virtually all the nucleated cells express class 1, in a broad sense, every nucleated cell presents antigen to the CD8 plus T cells. But professional APCs are the dendritic cells, macrophages, the B lymphocytes. So constitutively, it expresses the class 2 co-stimulatory molecules and the adhesion molecules needed to activate the naive CD4 plus T cells. Then you have some non-professional atypical APCs like the endothelial cells, the thymic epithelial cells, and some epithelial cells that can express the class 2 under inflammatory conditions, but it lacks full co-stimulatory capacity. And only professional APCs can prime the naive T cells. So without co-stimulation, the TCR engagement tends to induce energy rather than activation. So this is the key immune tolerance safeguard. So the key rule for T cell activation is that it should be constitutive, inducible MHC class 2. First of all, it should only the only professional APCs can prime naive T cells. So T cell receptor engagement alone without co-stimulation leads to functional inactivation or energy, right? And for the professional APC checklist, you can have constitutive inducible MHC class 2, co-stimulatory molecules, adhesion molecules. It should have the capacity to migrate to the lymphoid tissue and efficient antigen uptake machinery. Okay, now we will go one by one, like first with the dendritic cells. So this is the most potent professional antigen presenting cell. So immature cells, so it has a sentinel role. Like the immature cells resides in the skin and the mucosa, constantly sampling local environments. Then maturation and migration, where the pathogen recognition shuts down the antigen uptake and triggers migration to the lymphoid organs. Then there can be cross presentations, so certain subsets routes exogenous antigens onto the MHC class 1 molecules to prime the CD8 plus T cell responses against the tumors and the viruses. So in general, yeah, dendritic cells are links the innate and the adaptive immunity. They sample antigens in the peripheral tissues and undergoes maturation upon detecting danger signals and migrates to the lymph nodes to activate the T cells. The macrophages are has a somewhat different role. So the phagocytes doing double duty. So the primary role is to engulf and destroy the pathogens, debris, and the apoptotic cells via phagocytosis using lysosomal degradation. Then it plays a major role in the local antigen presentation by upregulating the MHC class 2 and the co-stimulatory molecules upon activation, reactivating the memory affected T cells at the site of infection. Then you have the Kupffer cells in liver, the alveolar macrophages in lung, microglia in the central nervous system. They have the same lineage tissue adapted phenocytes, and these are better. They are less efficient than the dendritic cells, but they're better suited to sustain and amplify an ongoing response rather than initiating one from the scratch. The B lymphocytes are antigen specific presentation via the B cell receptors. So the surface immunoglobulins or the B cell receptor binds antigens with high specificity and affinity. So this binding triggers receptor mediated endocytosis, and the uptake via the B cell receptor is far more efficient than the fluid phase uptake for the cognate antigens. Other part is the concentrates. It concentrates rare antigens. So because the B cell receptor mediated uptake is highly specific, the B cells can effectively present antigens. They function at antigen concentrations 100 to 1000 fold lower than what non-specific APCs require. It sets up the BT cell cognate help. So B cells presents peptides via the MHC class 2 molecules to the CD4 plus T follicular helper cells. This T cell reciprocally delivers the CD40L and cytokine help. So this interaction drives the antibody class switching and affinity maturation in the germinal centers. The constitutive class 2 expression, where along with the dendritic cells and the macrophages, the B cells constitutively expresses the MHC class 2 molecules, so no inflammatory induction is required for this expression. Then you have the thymic epithelium, Langerhans cells, and the inducible APCs. So the thymic epithelial cells includes the cortisol and the medullary. So it expresses both class 1 and class 2 constitutively, drives positive and negative selection of developing the T cells, and this is the most consequential antigen presentation in the body, shaping the entire T cell repertoire. The Langerhans cells are dendritic cell subset resident in the epidermis. It captures cutaneous antigen and migrates to the skin, draining lymphoid nodes. So basically, you need to remember what are the functions of these cells. That's all. It's not that complicated. So the endothelial and the epithelial cells are normally MHC class 2 low or negative, and IFN gamma, interferon gamma during inflammation can induce class 2 expression, and it's relevant to graft rejection and some autoimmune pathology. Follicular dendritic cells are non-hematopoietic, that they have a distinct lineage, so they retains intact antigen antibody complexes on their surface for B cell selection in germinal centers rather than the classical MHC presentation. So yeah, I'm not going into this summary. This is just kept in case you want to see it in one slide. I have put it for you. It's the same thing that I have been speaking on this. So I'm not going into the depth anymore. It's just a summary. A few questions on this particular part. So, which cell type is considered the most potent professional APCs for priming naive T cells? Yes, that's the dendritic cells. Okay. Which APC population is the most important for cross-presenting exogenous antigen to prime CD8 plus T cell responses against tumors or non-dendritic cell tropic viruses? Yes, that is the CDC1 or the CD8 alpha plus dendritic cells, which are specialized for routing phagocytosed antigen onto the MHC class 1 via cross presentation, critical for anti tumor and antiviral CD8 plus priming. Okay. The B lymphocytes are specially efficient APCs for antigens that. Yes. So the B cell receptor mediated endocytosis lets the B cell concentrate and present the cognate antigen at far lower concentrations than the non-specific uptake would allow. Cool. Now, which HLA class is constitutively expressed on nearly all nucleated cells, not just the professional APCs? So, class 1 is broadly constitutively expressed on every nucleated cell. Class 2 is restricted to professional APCs. The Bayer lymphocyte syndrome type 2 or the MHC class 2 deficiency results from defects in genes that are required for. So the Bayer's lymphocyte syndrome type 2 is a rare autosomal recessive primary immunodeficiency. and it is characterized by a severe absence of MHC class 2 molecule expression on the cells, and this genetic defect does not reside in the MHC class 2 genes themselves. Instead, it involves the mutated genes that codes for the transcription factors required to activate the expression of the class 2 genes, where it includes the MHC class 2 transactivator and the components of the RFX complex. So yes, answer C is correct. Okay, we go into the next part, where we will be talking about the antigen processing pathways. Okay, great. So the processing has two pathways. One logic match the antigen source to the display channel. So okay, so it's more of a antigen presentation comparison. You have the endogenous pathway and the exogenous pathway. The antigen source, so if we talk about the endogenous pathway first, where the antigen source is the cytosolic, self, viral, or the mutant proteins. It has a ubiquitin proteasome degradation system, the peptide transport through the tap 1, tap 2 from the cytosol to the endoplasmic reticulum. The loading component is the endoplasmic reticulum, where it has the peptide loading complex. The placeholder that blocks the premature loading, it's not applicable because the ER peptide pool is the intended source. The peptide editor is tepacin, and it's presented to the CD8 plus cytotoxic T cells. The exogenous or the class 2 pathway is extracellular. It has a phagocytosed material, that's the antigen source. It has an endosomal or lysosomal degradation proteases, the cathepsins. Peptide transport is not required. The loading component is the uh, M, so the MEC endosomal compartment. The placeholder that blocks the premature loading is the invariant chain to the clip. The peptide editor is the HLA-DM regulated by the HLA-TO, and it's presented to the CD4 plus helper T cells. Now, step by step of how we go from the from the endogenous pathway, that is the class 1 cytosolic pathway. So a little bit of step by step understanding. So first step is ubiquitination. So it happens in the cytosol. So action, it's a cytosolic self, viral or defective proteins are tagged with ubiquitin chains. This tag marks the target proteins for destruction. The proteasome degradation is occurring also in the cytosol, where the 26S proteasome breaks down the tagged ubiquitinated proteins. The proteins are chopped into short fragments known as the peptides, followed by tap transport, where it moves from the cytosol to the endoplasmic reticulum lumen. So the tap 1, tap 2, which is an ATP dependent transporter system, shuttles the short peptides across the membrane, and the peptides travel from the cytosol to the endoplasmic reticulum lumen. Followed by, there is the peptide loading complex in the ER lumen that facilitates the assembly. So chaperone proteins coordinates the binding, and the transported peptide is loaded onto the nascent class MHC class 1 molecule. Then from the ER, it goes to the stable MHC peptide complex exits the ER to the Golgi apparatus to the cell surface. So this complex is presented externally for recognition by the CD8 plus T cells, or the cytotoxic T lymphocytes. So the degradation occurs in the cytosol via the proteasome, loading occurs in the ER lumen via the PLC and the TAP transporters. The class 2 pathway is the vesicular endocytic pathway. So there are five main steps here. So first is the uptake, where the extracellular antigen is internalized by the phagocytosis, endocytosis, or the receptor-mediated uptake. Then the acidified endosome lysosomal proteases cleaves the antigen into peptides, leading to the endosomal degradation. In the ER, the class 2 alpha beta assembles with the invariant chain, which occupies the groove, so no ER peptide binds prematurely. Then there is the clip exchange, where the um, invariant chain is degraded to clip in mic vessels. The HLA-DM catalyzes the clip release and loads the antigenic peptide. The HLA-2 edits, regulates the DM. And then there is stable peptide class 2 complex traffics the surface for the CD4 plus T cells. So the question is, why the invariant chain matters? So class 2 molecules folds in the same endoplasmic reticulum as the class 1, surrounded by the pool of tap delivered cytosolic peptides. So this invariant chain physically occupies the groove during the ER transit and directs the complex to the endosomal compartment, preventing the class 2 from presenting the wrong cytosolic peptides and ensuring it samples extracellular endocytosed antigen instead. Coming to the cross presentation, where you have the loading exogenous antigen onto the class 1. So the dendritic cells engulfs external antigens like the tumor cells or the viruses that do not directly infect the antigen presenting cells through phagocytosis. So the internalized antigen enters a phagosome. So from there it can follow one or two pathways to converge on MHC class 1 presentation. The cytosolic route relies on the traditional cytoplasmic and endoplasmic reticulum protein degradation machinery. The internalized exogenous antigen is transported out of the phagosome into the cell cytoplasm. Then once it is into the cytosol, the antigen is broken down into shorter peptide fragments by the proteasome complex, and the resulting peptides are transported via the transporter associated with the antigen processing, the TAPS proteins, across the membrane and into the ER. Now, inside the ER, these peptides re-enter the MHC class 1 loading pathway, exactly like a endogenous antigen. The other route is the vacuolar route. So this route bypasses the traditional cytosolic and ER machinery entirely. So the exogenous antigen remains strictly inside the vacuolar system. The phagosomal proteases like the um, cathepsins directly degrades the antigen into the peptides within the phagosome itself. The MHC class 1 molecules are then recruited directly to the phagosomal membrane. The peptide loading happens entirely within the phagosome using ER derived machinery that has merged with the vacuolar compartment bypassing the main ER lumen. So, regardless of whether the antigen takes the cytosolic or the vacuolar route, both pathways ultimately converge on the same endpoint to the prime immunity, where you have the peptide MHC1 complex followed by exocytosis, cell surface presentation to the CD8 plus T cell primer. Right. So maybe try out these questions. In the endogenous pathway, which transporter moves peptides from the cytosol into the ER lumen? Yes, the TAP1, TAP2 forms an ATP-dependent heterodimeric transporter that delivers the proteasome generated peptides from the cytosol into the ER lumen for the class 1 loading. What is the main function of the invariant chain in the class 2 pathway? Yes. So the invariant chain physically blocks the class 2 groove during ER assembly and the Golgi transit, then routes the complex to the endosomal meek where it degraded to clip. Okay. The HLA DM's primary function in the class 2 pathways too. So basically what is the function, the primary function of the HLA DMs in the class 2 pathway? That's right. So the HLA-DMs acts as a peptide editor in the MIC, catalyzing the clip release and favoring loading of stable, high affinity antigenic peptides. The HLA-2 modulates the DM's activity. Next, what do you mean by cross presentation? Yes, that's correct. Cross presentation led specialized dendritic cells load phagocytosed antigen onto class 1 via either a cytosolic or a vacuolar route, enabling CD8 plus priming against antigens the DC did not synthesize itself. The next one says which component of class 1 peptide loading complex directly bridges TAP to the empty class 1 molecule. Yeah, fantastic. Tepecin, it physically links the TAP to the nascent class 1 or the beta 2 heterodimer, optimizing the peptide loading. The calreticulin and the ERP57 are additional chaperones in the same complex. Okay, so I think this is the last part of today's lecture, which is T cell recognition and the clinical relevance. Let me just check. Is it so? Yeah, so this is the last section. That's good. Okay. So, right. So the T cell APC interaction, receptors and the co-receptors. So what you need to know over here is that you have an adhesion pair where the LFA1, the lymphocyte function associated antigen, you have the APC ligand, which is the ICAM1, and this adhesion pair stabilizes the physical contact and prolongs the interaction time between the two cells. Followed by a co-receptor adhesion pair, where you have the T cell receptor in the form of CD2 LFA2, the APC ligand, which is the CD58, and the LFA3 or the LFA3, and it assists in adhesion, and enhances T cell sensitivity to the antigen stimulation. For the antigen recognition, the T cell receptor, you have the TCR plus the CD3 complex, the peptide MHC complex, the major, which is the major histocompatibility complex. It determines antigen specificity. The TCR reads the specific foreign peptide nestled inside the host MHC scaffold. The co-receptor binding, where the CD8 or the CD4 uh, is there, which is the T cell receptor. The APC ligand is the HLA1 or the HLA2, and it enforces MHC restriction by binding to invariant regions of the MHC molecule, bridging the internal signaling machinery closer together. So co-stimulation is the T cell receptor, where you have the CD28 the APC ligand B7, the CD80 or the CD86. So essential, it's so the main function of this co-stimulation is that it is essential for the full T cell activation, proliferation, and survival. So without this signal, the T cell becomes non-responsive and energetic. Now the MHC restriction, which is the Zinckernagel and the Doherty principle. So the cytotoxic T cells from the virus-infected mice could only kill target cells sharing the host's MHC haplotype, meaning that the T cell receptors read the peptide and the MHC molecules together rather than recognizing the peptide alone. So one Rolf Zinckernagel and Peter Doherty in 1996 Nobel Prize got the Nobel Prize in Physiology, Medicine for explaining graft rejection and the cellular immunity. Next, the HLA in organ and stem cell transplantation. So HLA mismatch drives graft rejection. So the recipient T cells recognize the donor HNA molecules as the foreign or direct allorecognition, or recognize the donor HLA-derived peptides on the recipient's APCs, which is the indirect allorecognition. The matching priority is that the HLA-A, B, and the DR loci are typically prioritized for solid organ and hematopoietic stem cell transplant matching. Closer matches correlates with the better graft survival and lower risk in HSCT, which is the hematopoietic stem cell transplantation. High resolution DNA typing is now a standard. So from evolution point of view, given that around 45,000 named alleles are there, so serological antibody-based typing has been largely replaced by sequence-based typing for accurate matching. And complete matching is rare outside relatives, so this drives the importance of unrelated donor registries and increasing HLA matched transplant protocols with enhanced immunosuppression. From the HLA and autoimmune disease point of view, associations. So the HLA-B27 is strongly linked to ankylosing spondylitis via proposed mechanisms like the arthritogenic peptide presentation or heavy chain misfolding that has been shown in quite a few publications in recent time. So specific alpha beta combinations appear to correlate with genetic susceptibility to type 1 diabetes by influencing the thymic selection. And the HLA DRB1, it features a shared sequence motif in the peptide binding groove that links to the rheumatoid arthritis risk and the severity. So the allelic variation reshapes the peptide binding groove, which changes which self peptides are seen during the thymic selection. That is one, and which self or microbial peptides are displayed peripherally. So both can tip the balance towards the autoreactivity. Now, HLA infectious disease susceptibility and drug hypersensitivity. Now, pathogen resistance or susceptibility, where certain HLA alleles correlate with differential control of HIV, hepatitis B, hepatitis C, and malaria. So, the heterozygote advantage at HLA loci is a proposed evolutionary driver of HLA diversity. HLA-B and above-severe hypersensitivity. So it's a classic pharmacogenic example. So this allele presents the drug or a drug modified self peptide in a way that triggers a severe T cell mediated hypersensitivity reaction, which is now a mandatory pre-treatment screening test. Another actionable pharmacogenic association is the HLA-B some and the carbamazine induced Steve Johnson syndrome. It's particularly relevant in the Southeast and South Asian populations. And the HLA genotype is now directly actionable in clinical prescribing decisions, not just a transplant matching curiosity. So these are some of the clinical relevance in the context of infection and pharmacogenomics. Okay, so a little bit of few questions on this. So MHC restriction as described by Zinkernagel and Doherty means that yes, so they showed that the CD8 plus T cells recognize a composite of peptides plus self MHC, not peptides in isolation, explaining why the cytotoxic T cells could only kill infected targets sharing the host's MHC haplotype. Which T cell co-receptor binds HLA class 2 and helps restrict CD4 plus T cells in recognizing the class 2 presented peptide. Yes, so CD4 binds an invariant region of the class 2 beta 2 domain, reinforcing MHC restriction and stabilizing the TCR engagement for the helper T cells, and CD8 plays the analogous role for class 1. In the two signal model of T cell activation, CD28 engagement with B7 on the APC provides. So the T cell activation requires two distinct signals to prevent inappropriate immune responses. Signal 1, which is the antigen specificity, so it occurs when the T cell receptor binds to a specific antigen peptide presented by the MHC molecule on a APC. And co-stimulation of the signal 2 occurs when the co-stimulatory molecules interact. So specifically the binding of the CD28 on the T cell or to B7, the CD80 or the 86 on the APC delivers this vital secondary signal which is required for the full T cell proliferation and survival. So it is the co-stimulation signal. Great. Now, HLA typing matching is most clinically critical for which of the following procedures? So what does HLA typing do? It matches the tissue antigens between the donors and the recipients. So close HLA matching prevents the recipient's immune system from recognizing the transplant as foreign and destroying it. It is critical for solid organs like kidneys, hematopoietic stem cells, like the bone marrow transplant to ensure long-term graft survival. So yes, option B is correct. However, blood transfusion is via a standard protocol where it relies on the ABO and the RH blood group typing, not HLA typing. And routine vaccination, it triggers the protective antibody responses, not for tissue matching. Antibiotic selection is also something not with human genetics. Okay, so that's the last question. Why are certain HLA alleles strongly associated with immune diseases like HLA-B27 with spondylitis, then the type 1 diabetes? So the genetic variations primarily change the amino acid sequence inside the peptide binding groove of the MHC molecules. Now this structural change alters which specific peptides, either from foreign microbes or its own tissues, are displayed to the T cells. So if an HLA variant accidentally presents a self-peptide that looks like a foreign thread, it activates an autoreactive T cell, triggering the autoimmune diseases like what has been mentioned, the uh, ankylosing spondylitis or the type 1 diabetes, things like that. So yes, because HLA polymorphism is concentrated in the groove lining residues, different alleles presents different peptide repertoires, altering thymic selection and peripheral presentation of self antigens, which can predispose to autoreactivity. So, right, what are the key takeaways? So, the first thing is that one locus, three regions, two presentation channels. Understand that structure dictates the function. A closed groove fixes short peptides, open groove accommodates longer variable peptides. Then the two parallel but mechanistically distinct pathways, APCs, are not equal. Dendritic cells prime naive T cells, macrophages largely reactivate or support existing responses. Thymic epithelium sculpts the T cell repertoire itself, and the HLA genotype has direct clinical stakes. These are some of the references which I've put up. Yeah, so that's all from my part. If you have any questions, you can ask me. So over here, some of the references are mostly of a few publications, majorly. Some old publications, some from a few books. Okay. So you can go through a few of these references if you want to study about these in more details. Okay, so another thing before I might miss out, like there will be no class on the 4th of September, which is a Friday, and also there will be no class on 11th September. So these two Fridays there won't be any class. Um, but the September 4th class has been preponed to Sunday, which is 30th of August, from the same time, 6 to 8 p.m. And so that will be the class of September 4th, which will be on August 30th. And there will be a class on 15th September, which is a Tuesday, from 6 to 8 p.m. instead of September 11th. Right, so I won't be taking a class on 4th and 11th. However, these classes will be taken on 30th August and 15th September respectively. So one has been preponed, one has been postponed. Right, so okay, so fine. Okay, great. Thank you very much then for joining. We will meet again in the next week.